If humans were changing genetically right now, what would you actually expect to notice? A new trait becoming common? A mutation appearing in more families? Some inherited differences quietly becoming more common from one generation to the next? Or could evolution be happening while the average person looks almost exactly the same? That is the first problem. A gene can become more common even when it provides no obvious survival or reproductive advantage. A population can change for several reasons, and the explanation that looks most obvious from the outside is not always the one its DNA supports across generations. Natural selection is only one possibility. Chance can shift gene frequencies, new mutations can appear, and old pressures can weaken without producing any dramatic change you would notice in a mirror. That is why Framingham researchers have tried to separate inherited traits from reproductive outcomes, while Pingelap shows how a small population can be reshaped by chance alone. Neither case gives us a crystal ball, which is inconvenient but scientifically useful. Before we ask what humans are becoming, we need a harder test. What would actually count as evidence that evolution is happening now? That harder test begins by separating a visible change from an inherited one. Suppose the average value of a trait shifts across a population. That tells us something happened, but it does not yet tell us what caused it. Nutrition, disease, education, prenatal conditions, or other parts of daily life can move a trait without changing the genes passed to the next generation. A useful modern example comes from Framingham, Massachusetts. The Framingham Heart Study began in 1948 and eventually followed thousands of people across multiple generations. Decades of family records let researchers ask questions that a single snapshot cannot. Do people differ in a trait? Is some of that difference inherited? And does the inherited part relate to how many children they leave? Evidence for selection becomes stronger only when those pieces begin to line up across generations. Even then, inheritance is not enough. A trait can run in families without being favored, and it can correlate with family size without causing that difference. Researchers still have to connect inheritance with reproduction across generations. A changing population is evidence of change. It is not automatically evidence of why it changed. That distinction sounds fussy until two completely different causes produce the same result on paper. One may involve natural selection. Another may involve nothing more deliberate than chance. So, if a genetic version becomes more common, the next question is not simply whether it helped. The better question is, what else could have moved it? Chance is easiest to underestimate when a population becomes very small. On Pingelap, a remote atoll in Micronesia, an inherited condition called achromatopsia became unusually common. People with it have little or no color vision, reduced visual sharpness, and strong sensitivity to bright light. Estimates have placed the affected share around 4 to 10%, while roughly 30% of the population may carry the related allele. Nothing about achromatopsia suggests an obvious survival advantage. The key event came in the late 18th century, when a powerful typhoon devastated Pingelap and left only a small number of survivors. Those survivors did not contain every genetic version that had existed before the disaster. They carried only a sample. That changes the starting odds. When their descendants repopulated the island, rare alleles present in that small group could become much more common simply because the starting population was tiny and isolated. Geneticists call this a founder effect, one form of genetic drift. The lesson is easy to miss because the result can resemble adaptation from a distance. A gene rises in frequency. A distinctive trait becomes common, yet no environmental advantage is required. Sometimes a disaster leaves such a small genetic sample behind that chance alone can reshape the generations that follow. Chance can reshuffle variation that already exists, but it still needs variation to work with. The next question is where new genetic differences come from in the first place. Every child receives DNA copied from two parents, but copying is not perfectly exact. A small number of genetic changes appear in the child that were not detected in either parent. These are called de novo mutations. Most will never become important, but they are one source of genuinely new variation. They are fresh changes, not copies of differences already circulating in the population. Researchers in Iceland have been able to count them directly. In 2012, scientists sequenced whole genomes from 78 parent-offspring trios and found a strong relationship between a father's age at conception and the number of new mutations in the child. In that study, each additional year of paternal age was associated with about 2 extra mutations. A later Icelandic study examined 1,548 trios and confirmed that the ages of both parents contributed to the number of new mutations, with the paternal effect substantially larger. Older parents do not automatically produce more evolved children. Most new mutations go nowhere. Mutation only supplies variation that selection or chance may later act on. Modern behavior can therefore affect more than who reproduces. Changes in parental age can also alter how much new genetic variation enters a generation. Now the puzzle becomes harder. Selection, drift, and mutation can all change what we observe over time. So when the same genetic shift appears in a population, how do we tell which cause actually moved it? A rising allele frequency is not the verdict. Selection, drift, and mutation can all leave a population with more copies of a genetic variant, but they get there in different ways. Selection becomes more plausible when an inherited difference consistently affects who leaves more descendants. Drift needs no advantage at all. It comes from random sampling, and its effects become easier to see when populations are small. Mutation plays a different role again. It can create a new variant, but it does not decide whether that variant will disappear, remain rare, or eventually become common. So the observation may be simple, while the explanation is not. To narrow the cause, ask who reproduced, how large the population was, and whether the variant was new or already present. But there is another possibility. Sometimes variants remain because selection no longer removes them as strongly. We usually notice selection when something useful becomes more common. But selection also works by removing changes that carry a cost. If a genetic variant strongly reduces survival or reproduction, it is more likely to disappear. If that cost shrinks, the variant can remain even without becoming useful. Human smell genes offer an example. A 2013 study across 11 human populations found signs of weaker negative selection around some olfactory receptor genes. These genes help sensory cells in the nose detect odors. The finding does not mean humans are steadily losing their sense of smell. It suggests only that some changes in these genes may now carry a smaller evolutionary cost. So a genetic change can persist without ever becoming an advantage. If losing some function matters less than it once did, variants that reduce that function may linger longer in the population. Geneticists call this relaxed purifying selection. This is a very different kind of evolutionary change. Nothing has to become faster, stronger, larger, or more efficient. A biological function can simply stop being protected as tightly as it once was. And that leaves one more assumption to test. Even if selection is active, must the average trait move steadily upward or downward? Not necessarily. Sometimes selection can operate without pushing the average clearly in either direction. A trait does not have to move in one direction for selection to be present. Picture people spread from a low extreme to a high extreme. If those at both ends leave slightly fewer children than people nearer the middle, the average can stay almost unchanged, while the extremes slowly become less common. That is called stabilizing selection. It favors neither simply more nor simply less. Someone watching only the average could miss it entirely. A 2017 study published in PNAS used UK Biobank data to examine several forms of selection in a contemporary UK population. The researchers reported phenotypic evidence consistent with stabilizing selection across many traits, although the effects were generally weak. That is not proof that humans are being pushed towards some universal average body. Different traits have different causes, and environmental influence matters. A population can stay centered in the same place while the extremes become less common. That matters because popular descriptions of human evolution usually look for movement: taller, shorter, earlier, later, more common, less common. But selection does not always announce itself by moving the average. Sometimes the change is in how variation is distributed across the population. So asking which trait humans are becoming may already be the wrong question. Selection can push, chance can wander, new mutations can appear, and old penalties can fade. The harder task is deciding which force left the change we see. Once those possibilities are on the table, the usual question about human evolution starts to look backward. Spotting an unusual trait is easy. Explaining why it became common is the difficult part. A headline might say that humans are developing a new feature or that a gene is spreading. That is only the observation. Before accepting the evolutionary story attached to it, ask what the evidence actually establishes. The cases we have seen point to very different explanations: reproductive differences, chance, new mutations, weaker removal, or selection that barely moves the average. Their real value is not identifying a future winner. It is showing us what the evidence still has to explain. Is the change inherited? Does it affect reproduction? Is the population small enough for chance to matter? Did the variant recently appear? Has an old disadvantage become less costly? Could selection be acting on the extremes instead of shifting the average? That is a much harder standard than simply noticing that something became more common. It also makes exaggerated claims easier to recognize. Contemporary human evolution is therefore less about finding the trait than separating what changed from why it changed. The first can sometimes be measured directly. The second is usually much harder. And even after all of that, those answers still do not tell us what a human being in 2100 will look like. That leaves the future where good science often leaves it, partly unresolved. Human genetic change does not need a visible destination, and the strongest evidence tells us far more confidently what changed than what humanity is becoming. Migration, medicine, reproductive choices, population history, and random inheritance are all changing at the same time. We can measure inherited differences and watch some genetic frequencies move. What we cannot know is which environments, behaviors, and reproductive differences will still matter decades from now. That uncertainty is not a weakness in the science. It is part of the answer. Evolution does not have to produce a visibly new kind of human for populations to keep changing genetically. And when a change does appear, the hardest part is often not detecting it. It is proving why it happened. So the most interesting question is not what people will look like. It is which changes happening around us today will still be detectable many generations from now.